Right, hello everyone. Today we're going to be talking about chromosomes in both eukaryotic, and we'll talk about how DNA is packed in prokaryotic cells as well. So if we focus on eukaryotic cells to start off with, we have two different ways that you'll see chromosomes represented. Okay, so if we look at this first diagram here, we have one chromosome, and this is also one chromosome. So they're both showing one chromosome, but the only difference is the one on the right has two sister chromatids. Okay, so it's been duplicated. But if the cell is not undergoing mitosis, then it will just exist as a singular form here. Now, the thing that makes it a chromosome is the presence of a centromere. So a centromere is this thing in the middle here. It connects the two sister chromatids, and it also exists in a one chromosome in this rod-like shape here as well. You'll still find a centromere. Okay. Now, you'll also see chromosomes represented in a diagram like this, and this is called a karyogram or karyotype. So you can see here that we have these two rod-like structures here. So often people get them confused as one chromosome, but what I've just highlighted is actually two chromosomes. Okay, so we've got a chromosome from, so this chromosome will be from the mum, and then this chromosome will be from the dad. Okay, so in eukaryotic cells, the chromosomes exist as homologous pairs. So that just means that one chromosome is inherited from each parent, and they contain the same genes in the same position. All right. Now, if we go back up to this one, so now we're just looking at one chromosome. All right. So if we're looking at one chromosome, there will be sections on this chromosome that will code for different traits. Okay. So on this one, that section of DNA might code for eye color. Okay. Whereas this section of DNA might code for hair color or something like that. Okay. And those different sections of DNA, they're called genes. Okay. So along the chromosomes, there's specific positions, and they're called gene loci. Okay. Where particular genes are located. Now, different versions of the same gene found at these loci are called alleles. Okay. So what does that mean? So if we go down here, so remember each one of these little rods is a different chromosome. So we have an allele here. Okay, so that section, that is a gene loci. And then on this chromosome is another one. Okay, but they will both code for eye color. Okay, so they both code for the same trait, but they code for a different eye color. So this one might code for brown. So that one might code for brown eyes, whereas this one might code for blue eyes. Okay, so that's a good example for alleles, different versions of the same gene. Okay, now at the end of chromosomes, you'll find these things called telomeres. All right, so telomeres are the repetitive DNA sequences that protect the chromosome from damage. Okay, and they also prevent the loss of important genetic information during replication. Okay, so they're protective things that go on the end of these chromosomes to protect the genetic material. Okay. All right. Now, the centromere, it also holds the chromatids together and plays a key role during cell division. So here, it's holding these two chromatids together. Okay. So now we'll have a look at this diagram here. Okay. There's one more thing that you need to be familiar with for these chromosomes and the way the DNA is packed, is these histone proteins are also very important. So the way it goes is this DNA exists in the double helix structure, right? And it will start to kind of coil and fold around these histone proteins here. So you can see here that the DNA is wrapping around this protein and it'll curl around it. And it's just the first step of it kind of condensing. Okay. Now, these histones with the DNA wrapped around it, they form a structure known as chromatin. Now, chromatin is kind of like a chromosome, but it's not as tightly packed. Okay. So a chromatin is a chromosome without the presence of a centromere. Okay. All right. Now, this allows large amounts of DNA to be efficiently organized and regulated within the cell. Okay. Now I'll have a look at prokaryotic cells. They're a little bit different. Now, prokaryotic cells have a different chromosome structure. Instead of having those linear chromosomes, they contain a singular circular chromosome that doesn't really have a nucleus. So you can see this red squiggly thing. That's actually the chromosome. Now, it's a circular chromosome, so it's a bit different in prokaryotic cells. It's not enclosed within a nucleus. And what's interesting is circular DNA is also found in mitochondria and chloroplasts. Okay, so it can be found in plant cells and animal cells as well. But the difference between prokaryotic and eukaryotic chromosomes is that prokaryotic is a single circular chromosome that is not enclosed within a nucleus. Okay, now there's also small extra loops of DNA, and they're called plasmids, and they can be found in prokaryotes as well. Plasmids often carry useful genes, such as those for antibiotic resistance. They can also be transferred between cells as well. So you can see here the plasmid here. Oops. The plasmid is here. Okay. So that's just a small extra bit of circular DNA. All right. Hopefully that makes sense. Quick summary for the different types of chromosomes in eukaryotic and prokaryotic